Hello, my name is Ioanna Petta and I'm from Greece. I'm a postdoctoral scientist in the Host Microbiota Interaction Lab of Professor Laureate at UZ Ghent and VIB, where we study disease mechanism and novel therapies for colorectal cancer. CRC is the third most commonly diagnosed cancer worldwide, with almost 2 million new cases per year and with very high mortality rates. The disease affects the colon and the rectum, the distal parts of our digestive tract, and it can metastasize to other organs such as the liver and the lungs. Today, most patients are treated with classical chemotherapy, radiotherapy, and surgery, but there is a need for more effective treatment options. One of the limitations to discover and test new therapies is the lack of representative experimental animal models that resemble the human disease and take into account the role of the gut microbiome. The term gut microbiome refers to the trillions of tiny organisms living in our gut, including bacteria, fungi, and viruses. Remarkably, we have roughly the same number of bacteria as the total number of cells in our body. These microbes are beneficial for our health as they help with food digestion and protect us against invading bad microbes. Importantly, a well-balanced microbiome is crucial for the development and correct functioning of our immune system. Our gut microbiome is strongly influenced by our dietary habits, as our food constitutes the food of the microbes living in our gut. Fiber-rich food, for example, supports the harmony of our gut microbiome and has positive effects on our immune system. In contrast, a Western diet containing lots of sugars, lipids, and red meat can disrupt the balance and allow enemy microbes to become dominant, causing inflammation. In many inflammatory disease and cancer patients, the composition of the gut microbiome is different compared to healthy individuals, and increasing evidence indicates that some bacteria can stimulate the initiation and progression of these diseases, including colorectal cancer. In order to unravel the mechanism by which specific bacteria regulate CRC development and therapeutic responses, our lab develops novel mouse models using germ-free mouse technology. This means that we can generate completely sterile mice and then introduce specific bacteria of interest. With these unique models, we can study the involvement of specific microbes in the development of CRC and its treatment challenges. The recent development and implementation of the so-called immune therapies has created new hopes for cancer patients. These therapies interfere, reprogram, and enable our own immune system to recognize and attack cancer cells and are very promising for the treatment of different types of cancer, including CRC. Unfortunately, the majority of CRC patients don't respond to immune therapy because of the complex interplay among the cancer cells, our immune system, and the gut microbiome. Of note, new scientific data indicate that specific bacteria can improve the response to immune therapy in CRC. In our unique mouse models, we can investigate this interplay and manipulate the microbiome composition or certain immunological factors to better understand their importance in the response to immune therapy. We hope that our research will open new perspectives for the development of more effective therapies in CRC based on combined microbiome and immune interventions. In summary, my research focuses on improving the efficacy of immune therapies for CRC treatment using the unique experimental animal models we have developed and keep on developing in our lab with the help of the Greek funding.